few, oh, we got it running? This is important. Go over a few ground rules for those of you that don't know about the procedures at UCSD for thesis defense. So I'm going to make a brief introductory set of comments. Then Alex is going to talk for about 40 minutes or so. Then we're going to open it up to questions from the general audience for as long as it takes. Then we're going to ask everyone but the committee to leave. And then the committee will ask Alex some questions. And Alex might disappear, the committee will deliberate, and then we'll come up with a plan moving forward after that. Okay, well, it's a real pleasure to introduce Alex Buckner, student in the lab for a while. Some of you might not know Alex's background. Oh, hey, Bruce. Alex came to my lab after deciding that maybe lab science wasn't for her and wanted to get more into data science. So she came to my lab with minimal kind of experience managing massive data sets of the type that other people in the lab have analyzed being in my lab. We talked about it, and I felt she had a skill set that could carry over from the lab to kind of data science-oriented research. So we made a plan, and that plan was to attack what we thought was a problem that was beneath the radar screen, most labs focusing on cancer, which is to consider the role of the germline, not in cancer susceptibility, but in tumor phenotypes. We thought we'll find some data on mutation rates in tumors, relate germline variants to it. This will be a unique angle. Little did we know two things. One, the data sets that we wanted to use were woefully incomplete. So Alex had to go back and recall all the variants in the germline in the TCGA database. That was a huge undertaking, and she'll talk about her experience doing that. The second issue was we kind of incorrectly assumed that no one was going to be interested in the topic, since after we started the project, we learned that the entire TCGA group had a committee Olivier knew about that was working on germline variants in cancer, which meant that Alex was going to now compete with about 40 other people around the states working on the same problem, and came away, I think, admirably, as you'll see. So she was able to compete with a very large number of other people. So she's going to tell us a little bit about that experience, talking about the role of germline variation in cancer. So Alex, take it away.
You guys hear me okay? If I stand here? Great. Cool. Thank you for the introduction, Nick. So like you said, today we're going to be talking about the role of germline variation in cancer. And the reason I've chosen such an ominous title slide and ominous title is because I want to draw attention to the fact that germline variants are an underutilized and underappreciated resource in these data sets. So what most people do in cancer genomics are interested in are somatic mutations. And these are genetic alterations specific to the tumor tissue. And in this image, they would represent the tip of the iceberg. They're smaller in number, and it's much more clear what they may be doing to drive cancer development. In contrast, what I was interested in were germline variants. These are genetic variants present in every cell of the body. They far outnumber somatic mutations, and it's much less clear what they may be doing to drive cancer growth. So what I was aiming to do in my thesis work was to both integrate germline variants and somatic mutations to better understand how tumors develop and how they take on certain characteristics. So I'm just going to give you a quick overview of what I'll talk about today. I'm first going to give you the big idea of why I'm interested in germline cancer when many other people aren't, and then talk more in detail about three of my aims specifically. In the first, I identified a technical artifact in the Cancer Genome Atlas data. And for those that aren't cancer genomicists, the Cancer Genome Atlas is the biggest public cancer data set available to people that data mine like I do. In the second aim, I looked for associations between germline variants and different tumor characteristics or tumor phenotypes. And in the final aim, I developed a novel method of phasing germline variants that takes advantage of some unique properties of cancer samples. So when I think about what the germline might be doing in cancer, I like to think about it in terms of the environment shaping growth. So here I'm showing examples of three very different environments. And no one would be surprised to know that these different types of environments support different types of life. So for example, palm trees can grow where it's hot and dry. Pine trees can survive in the cold, whereas oak trees require something more temperate. And obviously here I'm talking about the environment in terms of climate. But what if instead I was talking about the environment in terms of genetics? So every one of us has thousands of germline variants that act together to create this unique genetic environment within us that makes us an individual. And just like you could use information about what the weather is like outside to predict what type of trees will grow, what I was aiming to do with my thesis work was to use information about this germline genetic environment to predict what type of tumors would grow in different individuals. So in other words, I was looking for associations between germline variants and different tumor phenotypes. And when Nick and I first talked about this, as he mentioned, it seemed like such an intuitive idea, I wondered why someone else hadn't worked on it already. And we quickly came to realize that was because germline variant call data was not available from these large public data sets. So the data I worked with and everything I'll show you today came from the Cancer Genome Atlas. And while they make raw germline sequencing data available, they don't give you the nice curated final variant call set. And that's mainly to protect the privacy of the patients in TCGA. So if I wanted to ask these biological questions I was interested in, the first thing I was going to have to do was to generate my own set of germline variant calls. And so that leads me into my first aim, where I identified a technical artifact in the germline sequence data from TCGA. And before I dive too deep into this aim, I wanted to touch on something I mentioned on the title slide, which is the difference between a germline variant and a somatic mutation. So what I'm highlighting here is an example of a germline variant. This is just a position where an individual differs from the reference genome. So you can see here the reference genome is a G, and this individual's normal or germline sequence is a C. So this would be a G to C germline variant. And something I want to mention while I'm here is that germline variants are present in every cell of your body. And every tumor starts off as a normal cell. So therefore, every germline variant is also present in every cell of the tumor, which is what I'm demonstrating here. This G to C variant will also be present in the somatic sequencing data. In contrast to this germline variant, this is an example of a somatic mutation. Here, the reference genome is a G. This individual's normal or germline sequence is also a G, and there's a T that's specific to the tumor tissue. And something also I want to point out while I'm here is both germline variants and somatic mutations are present in the somatic sequence data. So therefore, in theory, they have the same capacity to affect how tumor cells are going to grow and take on certain characteristics. And I'm sure most people here aren't that aware of what germline variant calling is, but you don't have to worry about it because I basically already explained it to you. So to get germline variant calls from an individual, you would take a normal DNA sample, which for a cancer patient would typically be a blood draw. You'll sequence that sample, align those germline reads to the reference genome, and just identify the positions where that individual differs from the reference genome. And it's obviously much more computationally intensive and complicated than this, but that's the basic premise. So for my project, I called germline variants from 10,000 samples from TCGA. And as Nick kind of alluded to give you an idea of the scale of this, at the time I published this paper, this represented the largest set of germline variant calls in a cancer cohort, and the only people that beat me was actually the TCGA working group themselves. So once I had these germline variant calls, I did some initial quality control, and everything looked like it was going to be great. So I then moved on and started asking some more biologically driven questions. So the type of germline variants I was interested in looking at are called loss of function variants. And these are variants that are predicted to be highly damaging. So they're things like stop gains or frame shifts that are predicted to completely destroy function of a gene. If you carry a loss of function variant, you're essentially a genetic knockout for that gene. So I used annotation software to identify these loss of function variants in my samples. And it's well known from previous work that the average individual carries around 155 of these loss of function variants in their exome. So as a second layer of quality control, I wanted to make sure that my individuals in my cohort also had around 155 loss of function variants. And what I'm showing you in this box plot here are these are the number of loss of function variants per person. And these are all the samples in my cohort grouped according to what cancer type they have. This red line represents the expected value of 155 loss of function variants. And it should be immediately visually obvious from this graph that at least 4 cancer types have samples that have far too many loss of function variants, more than you would expect. And keep in mind, this is all germline sequencing data, so you shouldn't see cancer type specific differences like you might if you were looking at somatic mutation data. And so specifically, for those that aren't familiar with the TCGA acronyms, this was colon cancer, leukemia, ovarian cancer, and rectal cancer. And what this indicated to me was that there was potential for a batch effect in my data. And so what a batch effect is variation in the data that's not due to biology, but due to some kind of technical artifact. So some kind of way the samples were prepared that's causing variation in your data. And the reason I thought of this is that TCGA is an extremely heterogeneous data set. The samples were collected over a span of 5 years at 3 different sequencing centers across the US. And they used many different sample preparation and sample collection procedures. So, for example, they used different exome capture kits, different types of Illumina sequencers. Everything was generated in a different way. So what I did is catalog all possible sources of technical variation I could find about these samples, and look to see if there was any kind of technical factor that might be correlated with this weird phenotype I'm seeing of having way too many loss of function variants. And to cut a long story short, I identified whole genome amplification of DNA prior to sequencing as the source of this issue. And so whole genome amplification is an unbiased method to amplify small amounts of DNA. So you typically would use this if you had a very small amount of input DNA and needed more to load on the sequencer. What I'm showing here in this graph, these are 3 of those cancer types where they have some samples with more loss of function variants than you should have. And now I've just had the samples split according to whether the DNA was whole genome amplified, here in red, or not before sequencing. And what you can see are that the samples where there was whole genome amplification used have significantly more loss of function variants. But really the key piece of evidence that convinced me whole genome amplification was the culprit here was there were 12 individuals where one blood sample was taken, split into half, into 2 separate samples. One of those samples was whole genome amplified and then sequenced. The other sample was just sequenced as is. So what I'm showing you in this box plot here, these are the same 12 individuals in each box. The only difference between the 2 is that their DNA was amplified in one instance and not in the other. So any variation you see between these 2 box plots has nothing to do with biology and is just entirely due to whole genome amplification. So this really convinced me that something about this amplification process is causing too many loss of function variants to appear in my final data set. And I don't have time to talk about this in detail, but I spent quite a long time looking into these loss of function variants to try to understand how they got there, what they look like, is there any way I can remove them from my data set? And the fun story in this is we actually ended up working with Roger Laskin, who invented the whole genome amplification method TCGA used, to come up with a mechanism. But despite all this time I spent understanding these variants, I couldn't find any way to actually specifically remove them from my data set. And the reason for this is that there's a lot of naturally occurring damaging variation that looks a lot like a technical artifact. So there's no way you can specifically remove the bad variants without throwing away a lot of good important data. And at this point you might be thinking, okay, this is interesting from a technical standpoint, but I'm a biologist and I don't care about this. So I wanted to close with a motivating example as to why biologists should care about these type of things, even if you don't love variant calling as much as I do. So I have just a very simplistic example of a type of analysis you might run using this data. So you might want to ask the question, does any germline variant, is having damaging variants in any gene in the germline associated with getting a specific cancer type over another? And I've highlighted this using a positive control example. We know that damaging germline variants in BRCA1 are associated with the development of ovarian cancer compared to other cancer types. So you can just make a very simple contingency table, and you would expect that if you ran a Fisher's exact test on this, you would see that there's a significant enrichment of BRCA germline variants in ovarian cancer patients. So I could just run this analysis for every gene in the exome, just trying to see if there's other genes associated with getting ovarian cancer. And so here I have the results from that analysis. Anything that's in red is significant above a Bonferroni multiple hypothesis correction. And so you can see, indeed, BRCA1 and BRCA2 are positive controls, are significant, but so are 600 other genes. And on the bottom here, I used an incredibly stringent filter to try to remove those artifactual variants. And even then, I'm still getting way more significant hits than you would expect with this type of statistical analysis. And so really, the problem here is that one of your covariates of interest, which is what cancer type a tumor has, is highly correlated with this confounding technical factor, which is whole genome amplification. So if you ever see interesting results in the ovarian cancer samples, you'll never know whether that's due to ovarian cancer biology or the fact that all of those samples were whole genome amplified. So any kind of analysis you run on the germline data in TCGA that compares one cancer type to another is confounded by this whole genome amplification artifact. And you might be thinking that this is a very contrived example I chose to make my point, but I will assure you that I've seen papers published in the literature using very similar methods and using germline data. So it's not so out of the box that I made this up to prove my point. Other people do these types of things. And the other thing I just want to highlight here is the fact that this is a very sneaky type of problem, that you can do your statistics entirely correctly and still come to the wrong conclusion because there was something you didn't know about deep in your data set. So really highlighting the importance of spending all this time doing quality control so you don't publish something that's incorrect. And so to wrap up this portion of the talk, I drew attention to some of the problems that could arise when working with large heterogeneous data sets. And at the time we published this paper, there really wasn't much out there that described just the amount of heterogeneity in TCGA. So I think that's a very valuable resource to other people using this data set. I also specifically spent a lot of time coming up with how whole genome amplification causes artifacts. And this has implications outside of TCGA as well. Many applications use whole genome amplification of DNA. And so my analysis can kind of serve as a warning for the type of artifacts that might arise from that type of analysis. And so as I mentioned, I spent a lot of time trying to filter these variants out and couldn't find a way to do it well. So for all the analyses going forward that I'll show you, I just removed the whole genome amplified samples. So at this point, I finally had a clean germline variant call set. This is like 2 years of banging my head against the wall, by the way, for those keeping track of this. So I finally had clean data, and I was so excited to ask these biology questions that made me come to grad school in the first place. So what I was interested in is looking to see, can germline variants influence a tumor phenotype? And when I say tumor phenotype, that's an incredibly broad and vague term. That can mean anything. How fast a tumor is growing, what tissue it's in, what kind of mutations it has. But I really wanted to narrow down on one thing specifically to study in detail. And what I was looking for in a tumor phenotype was something I could get just from somatic sequence data alone, something that was variable within and across cancer types, and something where there was a clear biological mechanism underlying how that phenotype occurs. And so the phenotype that checked all those boxes for me was microsatellite instability, or MSI. And what I've illustrated here, this is an example of a microsatellite region in the genome. It's a short repetitive sequence. And these sequences are difficult for DNA polymerase to replicate properly. The polymerase can slip and either add or delete a repeat from the sequence pretty easily. In healthy cells, there's specialized DNA damage repair proteins called the mismatch repair proteins that can specifically identify and correct these types of errors. What often happens in cancer is these mismatch repair proteins are somatically altered and no longer functional. That allows mutations to accumulate in these microsatellite regions. And what this ends up looking like is this microsatellite region will either grow or contract depending on the type of mutations it accumulates. So this process of expansion and contraction of microsatellites is called microsatellite instability. And part of the reason this was such an attractive phenotype for me to study is that someone else had already taken the time to quantify this in the TCGA samples. So what I'm showing you here is their data. And what they did is they took the entire exome sequence, just the reference genome sequence, and identified all possible microsatellite sites just based on sequence alone. And then for any individual's tumor, they said what fraction of those microsatellite sites exhibit instability in that person's cancer. So what I'm showing here is the phenotype I used for this analysis, which is the percent of unstable sites of all possible microsatellites. And here I've plotted that for different cancer types in the TCGA, and I'm illustrating that this phenotype is variable across and even within cancer types. And like I mentioned, it's well known that somatic alteration of these mismatch repair genes is associated with developing somatic MSI. So it just seemed like the next logical step to me to ask whether damaging germline variants in these mismatch repair genes might also influence levels of somatic MSI. And so I've highlighted this with a very simple example. Let's imagine you have two individuals. One inherently has excellent DNA damage repair, and this other person has some kind of an inherited defect in how well their mismatch repair proteins work. Let's then imagine they both get the exact same set of somatic mutations that causes them to get cancer. It's easy to see that you would think that this individual with inherited defect in mismatch repair would develop a tumor that had higher levels of somatic MSI than someone whose DNA damage repair worked very well. And this is an incredibly contrived example because in the real world, no two people ever develop the exact same set of somatic mutations leading them to get cancer. In real world data, people vary in terms of their germline variants, they vary in terms of their somatic mutations, and they also vary in terms of their levels of somatic MSI. So what I wanted to be sure to do was to account for somatic mutations when I was doing these analyses. So I'm looking for an association between a germline variant and a tumor phenotype, but I want to build a model that can account for somatic mutations so I don't falsely attribute something to a germline variant that could be explained by a somatic mutation instead. And for this reason, I chose to use a linear model for these analyses. A linear model allows me to include covariates in the model that will account for somatic mutations when I'm looking for an association between a germline variant and a tumor phenotype. And so I'll just walk you through this quickly. Essentially what I'm doing is trying to predict the amount of MSI in an individual's tumor, using information about what germline variants they have, what somatic mutations they have, and what genes are epigenetically silenced in their tumor. So what this linear model will allow me to do is determine how much variability in MSI I can attribute to germline variants and how much I can attribute to somatic mutations. So I use this model on a gene-by-gene basis. There are a list of 222 DNA damage repair genes, 27 of which are these mismatch repair genes where you would expect most of the signal to be coming from. And so here I'm showing you the results from that analysis. On the left here, I'm showing my results from the germline term in my model. So this is saying germline variants in these genes are associated with having somatic MSI. And this is the somatic mutation term in the model, so somatic mutations in these genes, MSI. And how you interpret these is anything that's in red was significant above a multiple hypothesis correction, and anything that's in this upper right quadrant, there's a positive association. So if that gene is altered, there's more MSI in that individual's tumor. So if we start by looking at the somatic mutations, my top hits are all known mismatch repair genes, which is great. Particularly my most significant hit is MLH1, which is most frequently identified as the cause of somatic MSI. So this is great because it gives me confidence that my model is reasonable, I can recapitulate true biology. So for that reason, I was very excited when I saw a significant hit from my germline term in the model. This would say that damaging germline variants in PMS2 are associated with higher levels of somatic MSI. And this was really great because at the time, there were very few published studies showing a direct association between a germline variant and a tumor phenotype. But being the perpetually skeptical person that I am, when I saw PMS2, the first thing I thought of was not, let's write this up as a paper, it was, uh-oh, what about Lynch syndrome? So for those that aren't aware, Lynch syndrome is a well-known cancer predisposition syndrome. And this occurs when individuals inherit a damaging variant and one of 4 mismatch repair genes. MLH1, MSH2, MSH6, and as I've illustrated here, also PMS2. And so what this looks like is these individuals inherit a germline variant in one copy of this PMS2 gene in every cell of their body. And this essentially makes them a ticking time bomb. If they acquire a second somatic mutation in the second wild-type copy of PMS2, those cells that acquire that somatic mutation rapidly become cancerous. And specifically, they'll turn into a tumor that has high levels of somatic MSI because the mismatch repair pathway is deficient. So really the key feature you're looking for in Lynch syndrome is this biallelic alteration. And so for Lynch specifically, it's a combination of an inherited variant and a somatic mutation that affects both copies of the gene. And I'll be talking more later about maternal and paternal chromosomes, so keep this in mind. So you might be thinking at this point, well, if you knew about Lynch syndrome in the first place, Alex, why didn't you just look for this when you started this project? And in my defense, and in defense of the TCGA community, everyone thinks of TCGA as a sporadic adult onset data set. So the median age of onset is around 60, and typically individuals with these types of Lynch syndrome would get cancer far earlier than that. So it just wasn't even on my radar as something to look out for. But obviously, getting this result, I thought I should be prudent and look for evidence of Lynch syndrome and TCGA. And as I'm sure you're aware from the foreshadowing on the title of this slide, I indeed did find evidence of Lynch syndrome and TCGA. And what I mean by evidence of Lynch syndrome is I found individuals carrying biallelic alterations in one of those 4 known Lynch syndrome genes. I specifically found 6 individuals with biallelic alterations. And what I'm showing you here in these box plots, these are all individuals in my cohort grouped according to how those 4 Lynch syndrome genes were altered. So in blue here, these individuals had no genetic alteration to those genes at all. These individuals only had germline variants. They only had somatic mutations. And these are those 6 individuals I identified with a biallelic alteration. And here, this is my somatic MSI phenotype. And so it's very easy to see that these biallelic alteration carriers have much higher levels of somatic MSI than almost everyone else in the TCGA. Further, I can show that these biallelic alteration carriers also have significantly earlier age of cancer diagnosis, which would be in line with the idea that they have a predisposition syndrome. Also, these 6 individuals all have a cancer type concordant with Lynch. They either have uterine or colorectal cancer. So really, all of the data I have on these individuals would indicate that they do indeed have Lynch syndrome. And this was very annoying for me at this point because this is the last thing I wanted to find in my data. And if I remove those 6 samples out of a cohort of around 7,000 people I was working with, and rerun that linear model, as you would expect, that PMS2 hit is gone. So it was, as I feared, I wasn't finding an association between a germline variant and tumor phenotype. I found an association between biallelic alteration and a tumor phenotype. And I won't have time to go into too much detail, but I did spend quite a bit of time then digging to say, okay, let's throw out the lymph samples. Do I find any evidence that germline variants can influence somatic MSI? And ultimately, I found no evidence of that. So I created a helpful little graphical abstract to help you remember these findings. So there were individuals in TCGA with just damaging germline variants, those with biallelic alteration, and it was only really the biallelic alteration carriers that you could see a somatic phenotype in. So I did this large unbiased analysis of the germline to rediscover the MINSIN2 hit hypothesis and rediscover Lynch syndrome. So for the people in the audience that are statistically inclined, you might think, well, hey Alex, if you had included an interaction term in that linear model, you could have saved yourself a lot of heartbreak. And I agree. So I did go back and try that. I was like, man, I should have done this in the first place. So for the less statistically inclined, when I say include an interaction term, essentially all I'm doing here is saying, do germline variants influence MSI? Do somatic mutations influence MSI? And then do biallelic alterations influence MSI? So I'm just creating a separate term to account for biallelic alterations. But what's actually interesting is if I run this model now, looking for biallelic associations, I don't get any significant hits. And the reason for this is there were only 6 people that had biallelic alterations. So for any single gene, I was underpowered. I couldn't find any significance. So it was really only using the biological prior knowledge that those 4 genes all cause Lynch syndrome and could be grouped together that I was able to find the signal in this data set. So this kind of harkens back again to the whole genome amplification kind of example to show how it's incredibly easy to come to the incorrect conclusion when you're working with big data sets, and to teach people to always be suspicious of findings that seem too good to be true. It's really worth your time to dig into those and make sure it's not some kind of artifact. So to wrap this up, I identified at least 6 individuals in TCGA that appear to have Lynch syndrome. And I didn't have time to talk about it, but I did look for other types of cancer predisposing syndromes and found evidence for up to 60 people that might have some kind of predisposition syndrome. And you might be thinking these are very small numbers. We're talking out of 10,000 people. But as I clearly demonstrated today, 6 samples were enough to utterly confound my analysis. So it's something that everyone should be aware of that's working with this data set. In addition, the big finding that I had that sort of flies in the face of the entire title of my thesis is that germline variation alone had very little effect on this tumor phenotype. I was studying somatic MSI. But despite this, I'm actually not that discouraged. I think my study was very limited in that I was looking at one single somatic phenotype. And as I mentioned earlier, I was looking at these germline loss of function variants. So there's many other germline variants I didn't include in these analyses. So I think the bottom line is I was looking for the low-hanging fruit, and I didn't find it. So whoever follows up on this study is going to have to reach higher. All right, and then for the very last portion of my talk, I'm going to switch gears a little bit and instead talk about germline variant phasing. So this is kind of like the life gives you lemons part of my thesis, where like I got all upset about Lynch syndrome, and it's like, what else can I use this data set for? And I ended up coming up with a really cool story. So I've been talking a whole lot about the germline today, but I've been simplifying things. So humans are diploid. That means you have 2 copies of every chromosome. One you've inherited from your mom, and one you've inherited from your dad. And in a typical high-throughput sequencing study, if you identify a heterozygous germline variant, you don't necessarily know whether that was on the maternal chromosome or on the paternal chromosome. And this idea of determining what chromosome a variant is on is called germline variant phasing. And for a single variant in isolation, it might not matter to you too much. But you can imagine a situation where you have 2 damaging variants in the same gene. And there's 2 outcomes here. Either both of those damaging variants will be in the same gene copy, and this is called cis phase, or you could have a damaging variant in each copy of the gene, and this would be called trans phase. And I've chosen to illustrate this with MLH1, which hopefully people remember from 3 slides ago, is a Lynch syndrome gene. So I've chosen that to illustrate the fact that this individual would have Lynch syndrome. They have 1 damaged copy of a mismatch repair gene, but the other is still wild type, and they would get cancer at an advanced stage. In contrast, this is actually a different type of predisposition syndrome called biallelic mismatch repair deficiency. And these individuals develop aggressive, very early onset pediatric cancers. So to illustrate the fact that knowing the phase of variants can be critically important and have an immense impact on an individual's phenotype. So as I was working with this paired tumor and normal sequencing data, I noticed something interesting. So if you sequence normal cells, all those cells should be diploid, and the DNA in your sample should be like half of it should come from the maternal chromosome, half should come from the maternal chromosome. And you can reasonably expect that your sequencing read should be 50-50 split between the maternal and paternal chromosomes. What often happens in cancer are somatic copy number alterations. So these are gains or losses of either whole chromosomes or portions of a chromosome. Here I've illustrated a whole chromosome gain of the maternal chromosome. There's going to be more DNA in that sample coming from that maternal chromosome because it's been amplified. And therefore, you would also expect that you'll see more sequencing reads coming from that maternal chromosome. And so let's go back to this idea of a heterozygous germline variant. We overlay that here. In your normal sample, a heterozygous germline variant is going to be on half of your chromosomes, half of your DNA, should be on half of your sequencing reads. The variant allele frequency of that variant you can expect should be around 0.5. But as I've mentioned many times during my talk today, all germline variants are also present in all cells of the tumor. So if we look for that same germline variant in the somatic sequence data now, you see something interesting. Now that variant looks like it's increased in allele frequency because that chromosome the variant was on has been amplified. So there's more DNA that carries this A variant and there's more sequencing reads that carry the A variant. So the variant allele frequency of this A site will now be 0.75 in the tumor. And what I realized is you can use information about changes in variant allele frequency between your normal and your tumor sample, which I'll refer to as delta VAF, to determine what chromosome a variant is on. So let's expand this example now to include multiple heterozygous variants. So here to A, this is exactly the same thing I just walked you through on the last slide. You can see that the delta VAF for this A variant should be increasing in the tumor. But in contrast, if we look at this heterozygous G variant that's on the paternal chromosome, this will appear to decrease in the tumor. So the delta VAF would actually be negative for the sample. And so what the delta VAF is telling you about the A variant is that it's on a chromosome that is relatively more abundant in the tumor sample than the normal sample. And what this is telling you is the G variant is on a chromosome that's less abundant in the tumor sample than the normal sample. And with those two pieces of information together, you can say that those two variants can't be on the same chromosome. So they must have been in trans phase originally in a germline, which means they were on opposite chromosomes. And so you can use information about the delta VAF changes of multiple variants on the same piece of DNA to determine the phase of those variants. So I've tried to illustrate this here with another cartoon. So on the left, this is this example I just walked you through. If you have germline variants in trans phase on opposite chromosomes and have a copy number alteration here, you expect one of those variants, the delta VAF, should increase, and the other variant, the delta VAF, should decrease. So when you see two variants with delta VAF changing in opposite directions, they were on opposite chromosomes. And the alternate situation, if you have germline variants in cis on the same chromosome, copy number alteration, you'll expect the delta VAF should change in the same direction for both of them. And so therefore they would be in cis phase. And I think it always helps to actually look at some real world data. So here I actually have the delta VAF for all the heterozygous germline sites along one chromosome for one TCJ sample. Here in the gray region is an area where there were no copy number alterations in the tumor. And so the delta VAF values here are centered around 0, as you would expect. There's no changes in chromosome number between the two samples, so the delta VAF for a germline variant should be the same between both of them. And in contrast here in purple, this is a region of copy number alteration. And to me, when you look at this plotted out, it's so much more visually obvious that all of these variants must be on the same chromosome, and that chromosome is increasing in frequency in the tumor, and then vice versa, these must all be on the opposite chromosome. And so I'm just going to give you a very whirlwind tour of this method. I call my method VAF phasing. So the input data I use is very simple. I just need reference and alternate read counts for all germline variants in a paired tumor and normal sample. Using this data, I can identify regions of copy number alteration. Because keep in mind, I can only phase variants in regions where there's been changes in chromosome number between the two samples. Once I identify what regions are copy number altered, I then determine what variants in those regions I feel confident enough to phase. And then finally, I output phase calls. So I'll talk about these two steps in a little more detail. So how do I identify copy number altered regions? So I realized you can actually use the absolute value of delta VAF to identify copy number altered regions. Because the delta VAF in a region without a change in chromosome number should be centered around 0. If there's been a change in chromosome number, it will be greater than 0. So you can use the absolute delta VAF value and feed that into an algorithm called circular binary segmentation. This is an algorithm often used to find copy number altered segments. Essentially what I'm doing is feeding in the absolute delta VAF value, and this will the algorithm walks along the chromosome and identifies segments with similar magnitude delta VAF values. So I've shown that here. Now I'm plotting the absolute delta VAF value, and these red lines are segments identified by the CBS algorithm. So according to this algorithm, this segment, all of these variants should have a mean value delta VAF that's the same. So you would say this is all probably one somatic copy number alteration event. Within any of these segments, I then want to determine what ones of those variants I feel confident to face. And what do I mean by feel confident? What I mean is, do I have sufficient read evidence to say that I think this change in VAF is actually due to a change in chromosome number and not just due to sequencing noise? So I assess this just using a Fisher's exact test and the read count data. So if I see a significant Fisher's exact test, I say that I think that's sufficient evidence that there's a chromosome change and it's not just undersampling of sequencing reads. So that's the method at a very high level. There's way more detail that goes into this if people have more questions later. But for the take-home message here, I applied this method to 7,000 samples from TCGA, along with 2 other standard computational phasing methods. These other methods are called read-back phasing. And how they work is they actually use the sequencing reads to phase. So they look for a sequencing read that spans multiple heterozygous sites. So if you see the same heterozygous sites occurring on the same read, you can assume that those sites came from the same chromosome. So this works incredibly well, but it's limited by the fact that it can only phase variants that sequencing reads can span. So you can really only phase at distances up to a few kb at most. And this is just due to limitations on Illumina insert and read size. So I ran those methods and compared them to mine, and my method was highly concordant with theirs. And in addition, I can actually phase on average 33% more variants by taking advantage of this copy number alteration type of phasing. So that's what I've illustrated here. Is these are the other methods, and this is with the addition of my method. And the interesting thing about this is the variants that are accessible to my method that aren't accessible to them are those that are far apart. So they're limited by the size of an Illumina read, whereas I'm only limited by the size of a copy number alteration, which, as I said, can be up to the length of an entire chromosome. So in theory, I can phase variants along the length of an entire chromosome. So to summarize this, I can phase more samples, more variants using this method. And I think VAF phasing would be a great add-on to a cancer informatics pipeline. I don't see it as something that's going to revolutionize phasing, but I think it's an excellent way to squeeze more data out of some of these existing data sets. So kind of, digging through the garbage and finding some gold. That's kind of how I feel about my TCGA data mining experience. So to wrap up everything, my PhD, I have drawn attention to numerous deficiencies in TCGA. But I know I've been really negative, and my lab yelled at me for this. 90% of TCGA is still good. The unfortunate fact is it took me 4 years to find the 10% that wasn't. So go forth and be fruitful, and don't make the same mistakes I have. And then overall, I think I did show that integrating germline and somatic data does add a lot of value. So I demonstrated this by identifying those Lynch syndrome samples and developing a novel method of phasing that no one had really thought of before. And to conclude, I'd like to borrow an analogy from Shrek and say, much like ogres, tumors are also like onions. There's multiple layers of genetic alteration that go into shaping a tumor. And like I've been saying so many times today, germline variants are present in every cell of the tumor. So they would be there at the core of the onion. And then on top of this, you develop things like somatic mutations or copy number alterations that just add layers of complexity. But it's really only through integrating all of this data that we're going to understand where tumors come from, how they grow, and how we can best treat them. And I also had to put this in at the request of my lab. There are future directions. I just won't be doing them. But if someone wants to take this project, what I would suggest, and I kind of hit on these already, is that I was very myopic in that I only looked at one tumor phenotype. There is a world of other things you can look at. Particularly for the genomics people, the mutational signatures is the hottest thing right now, and I think there's going to be novel biology there. I was also very limited in terms of what germline variants I was looking at. There is so much room to do more complex things that I did not. And also, I think there would be value in looking at other data sets. So pediatric cancer would be an appealing data set to look at. A, because we think germline is probably influencing tumor more just because they get cancer earlier. And B, because there's going to be fewer somatic mutations you have to account for that are going to confound your analysis. So that's all I'm going to say about science. But I left myself a lot of time because I have to say thank you to a whole bunch of people. So I've been super lucky to have a ton of help during my PhD. And this was actually my former lab mate, Kunal, told me it takes a village to get me through my PhD. So I'm going to start first and foremost with my family, because they are going to watch this tape later. I don't know if I didn't make it ours. So they have been listening to me complain about my PhD for a solid 6 years, and I bet they are so excited that those phone calls are and stuff. When I was trying to make a slide for my friends, I realized we hate taking photos. So in lieu of that, I'm just going to give you Grumpy Cat and say thanks to all my friends for keeping me sane. In typical grad student fashion, I wanted to use the most embarrassing photo of my PI I could find. So this is old St. Nick short. We're really got to get into the holiday spirit there. I'll keep it off of his email still. It's the naughty and nice list. That's this one. And he said this at the beginning, but it was kind of like a crazy thing I decided to do. Like, I was working in a wet lab and came to Nick with no programming experience, no statistics. I don't know if he knows this. I don't know how to type. So like, that's like how woefully prepared I was for working in an informatics lab. And he was just totally down and was like, sure, join my lab. Here's a computer, Alex, I'll figure it out. And it seems like a crazy thing for him to do, but I cannot thank him enough for giving me that opportunity. Like, this is an amazing thing I was able to do, that I could make this transformation from a wet lab scientist to a bioinformaticist, and that he let me join his lab and learn from him. It's just amazing. I'll never be able to thank him enough, and it's made such an enormous difference on my PhD life and my future career. It's hands down the best choice I've ever made. Everyone should learn how to program, guys. And as I'm talking about Nick, I would also have to thank the lab. We also hate taking photos. This is from 2014. Chris will remember this. So I have to shout out to former lab mates, one of which is here, Chris and Dan. And also Dan Jimmick-Corliss, who couldn't be here today because he's off working in real life in an industry job. But they spent so much time teaching me things in the lab and have taught me everything I know about statistics, everything I know about programming, and everything I know about baseball. And shout out to Kurt Sharklab, who I hope appreciates the fact that I photoshopped them into this picture. You can't even tell, right guys? And so, as you can imagine, switching labs in grad school is frowned upon, and a lot of people thought I was nuts, like rightfully so. But really, there were two people that totally supported that decision and cheered me on, or were super supportive. So I will call them my cheerleaders. That would be Arshad and Tracy. So they were so supportive to me at a time when it was like the pit of despair in my grad school moment. And if it hadn't been for them, I don't think I ever would have finished. So I know I've thanked them a million times, but I have to thank them again. They were just there for me when I needed them. So you guys are awesome. I'd also like to thank my committee, and particularly Hannah and Olivier. So I'm sorry, Hannah, we don't have a cute selfie together, so Olivier gets this one. They've been like my committee members, but like really they have gone so far above and beyond and been so generous with their time, mentored me about every possible question I've ever had. Like, I've just been so fortunate to be able to work with them. I would say that any bioinformatics grad student is only as good as their computational cluster and their sysadmin. So I have to thank NRMB and Barry, who is here today. Like, you have no idea the scale of the analyses I did was bananas, like for people that don't do computational stuff. And the only way I was able to do that was through NRMB and through Barry's help to keep me from burning NRMB down. I only burned it down like once or twice over the span of 4 years, which is pretty good. I also have to thank my grad program, Biomedical Sciences. I can't say enough good things. They've made me feel like so welcome here, and it's like my second family on the West Coast. I have to thank Craig Venter, which is where my butt actually sits these days. And particularly, I have broken 2 MacBooks over the course of my PhD, and they just keep supplying me more. So I bet their IT department is also really excited for me to get out of here. And last but not least, I have to thank my funding. So I was part of the genetics training program, and I complained bitterly about the 8 a.m. Journal Club. But in retrospect, it was an amazing training opportunity. So thank you, Bruce, and thanks for coming today. And then also, T-Gen has given me both money and collaborations, so they've also been incredibly helpful. And I'll finally stop talking and ask if anyone has any questions.
Great. All right, we'll open it up to questions from the non-committee members.
First of all, wow, I'm so impressed with you. I don't know anything about genetics, so I only have one question, and it's just going to be a softball. method that you came up with. I don't know if you, maybe this is not a polite question to ask, maybe if you could talk a little bit about the limitations. Like, could you take this method and run it through somebody's blood as a diagnostic to look for either early cancer screening or some other thing that would be affected by, biallelic mutations?
I don't think
Sickle cell.
no, I don't. For the second question, no. I would say for the limitations, I can only phase things that are in copy number alteration regions. So it works better for tumors that have a lot of aneuploidy. You can phase more variants. And also other limitations are how much sequencing depth you have. Because there is that problem of like, is this noise or is this actually a change in copy number? So the two biggest limitations on the method are how much copy number alteration there is and how much sequencing depth you have in the tumor and normal sample. And yeah, in terms of like a diagnostic test, I don't think it would ever be that useful. I think something else would always be the better choice. So I'm more so selling this as like, this would be just a way to get more out of an existing data set, like really easily. This is incredibly easy to implement and you just get extra fun stuff out of your data.
So thank you for sharing your limitation strategy for going through this because that would be valuable for us for working through the pipeline. But so just a general question then. So if you could go back 2 or 3 years to the beginning of your project, is there anything that you would do differently to go through the journey or change your project?
Yeah, I mean, it's hard because I'd say I'd choose a different phenotype, but I wouldn't know that at the time, right? Honestly, I don't think I could have foreseen any of the problems I had had. I think I would have done it the same way if I had done it again. Yeah, no, that's a good question. You're just asking because you know you're going to have to work on this. You're like, what should I do differently? I think the only thing I probably would have taken more time to do is spend more time thinking and looking at the data before I embark on a big adventure. So I just kind of like hit the variant column hard and didn't, I tell you, I cataloged sources of technical variation. Yeah, that happened afterward, right? Like I should have done that first, right? Like really spend time thinking about where my data came from, what are some possible things we might hit roadblocks before you start running 10,000 samples on NRV would be my advice, and probably also Gary's advice.
Great job. So I'm curious, so you found, I think it was 12 samples that someone had taken the time to sequence both the raw DNA and the whole genome amplified DNA. So they didn't notice that there was a problem.
So the thing is, the problem is specific to indels. So it was more noticeable to me, I think, because I was looking for loss of function variants, which are often indels. So I think if you just looked overall, you wouldn't see it, right? It was just the type of analysis I was doing. But I mean, a lot of people are looking at damaging variation, right? But I would also say at this point, TCGA actually found out around the same time I did that there was a problem. So going back to how Nick said, I'm basically been competing with TCGA. They found the same problem eventually, but it took them just about the same amount of time as it took me. So I think it really took like a really deep dive into the data to find it, and it took them quite a while to figure it out.
So, given your experience, is there, like, we have lots of genetic data nowadays, but it's really siloed. Is there value in aggregating it, or is it just sort of a fool's errand and you can't actually get rid of the technical variation in there?
Yeah, no, that's a question Michelle and I have been talking about, whether she wants to call germline variants with my cohort or have, separate little cohorts. I think the bottom line is you just need validation data sets. I think you can spend a whole lot of time trying to make the cleanest data possible, but at the end of the day, if you don't see it in a second set of data, it's meaningless. So I think you're probably better off having small testing and training sets instead of one giant data set, I guess is how I feel about that.
Really terrific talk. Particularly enjoyed the Lynch syndrome story. Really appreciate that you were able to dig into that to the depth that you did. But regarding the whole genome sequencing effect, was there a continuous variable that might have indicated the degree to how messed up things might have been that you tried to incorporate into your analysis to try to control for that effect?
I mean, probably if I had actually had all the data I needed. So all I got from TCGA, these samples have been whole genome amplified, those ones haven't. Like, yeah, I'm sure if I had like input DNA, like how many cycles of amplification, but like you don't have that data. So I think there would be a way. But it's just, once again, TCG is like missing data central. But that is a good idea, yeah. So, I mean, the other thing, what I've actually seen in a paper is someone has said we validated that they weren't whole genome amplification artifacts by looking by I and IgG. So you can also do that. Just got like 39 postdocs to help you.
Just so you know, everywhere is missing data central.
Yeah.
So
It's true.
forward, you just have to keep.
Yeah, this is for my life. This is my life now, okay?
All right, I'll just make a couple of quick comments because Alex is somewhat humble. But this experience looking at the QC and this artifact, we actually did put the paper up on bioarchives before we had it published, and it immediately got like thousands of hits and tweets from people basically saying they were so happy that someone went to the trouble to mine the data in the way Alex did. Another thing was in her assessment of the Lynch syndrome stuff, we actually had to debate whether we could publish the paper because there were so few people implicated in the data set that based on their characteristics, somebody could identify those individuals. So I actually spent some time on the phone with someone from Ned Sharpless's office at NCI talking about this issue. They acknowledged it, but they also acknowledged that they had no way of dealing with it. So they encouraged us to publish the paper with all the caveats. So the paper is actually being in review. So a couple of offshoots of the work that Alex performed. The last thing, just more of a humorous anecdote, because Alex mentioned her family. Her family came down to vacation in San Diego and spent some time. Of all the places they spent time, it was the unit above mine up in Solana Beach.
Did you recognize the furniture? That's where we are in that photo.
Literally
Nick
right
is,
above
Nick is right
mine.
downstairs.
Right
Yeah.
downstairs.
Yeah.
So all the places to rent.
I realized this after I told my family how often my PI is for like 2 hours, and we saw him outside.
True story. What a bizarre coincidence. Okay, I'm going to ask the non-committee members to go ahead and step outside.
When did they receive?